Hello and welcome students. In this video, we are going to talk about enzymes. The objectives, basically, the goals of this video are going to be a basic overview of enzymes or role of enzymes in metabolic pathways. The next one is enzymes as catalysts as to how they speed up or help with chemical reactions inside the body. And then next one, enzyme regulation factors. What factors affect enzyme activity? After this, I also want to give a thanks to OpenStack's Biology 2 ebook. They have shared their resources with me free of cost. So basically, all the pictures that are going to be used in this video and the notes are all from OpenStack's Biology 2 ebook. All right, so let us start by looking at the basic definition of enzyme. Enzyme is a protein. And a very important part is that it is a protein that functions as a catalyst. Catalyst is basically using to speed up a chemical reaction in the body. So they actually help facilitate the rate. The rate is really, you know, it makes it faster for the chemical reaction. The reactions will still occur in the body without an enzyme also. It's just that it'll take more time. So with the enzyme, it's faster. All right, now we are going to take a look at some of the characteristics of enzymes. Now we are going to take a look at the characteristics of enzymes, and we are also going to highlight the important words that are going to stand out about that characteristic. So the first one is that they are involved in every chemical reaction in our body. Next is that they are proteins. Now, what is the meaning of protein? Basically, they are um, a polymer of amino acid. So if these are monomers, amino acids, they are all combined together, joined together to form proteins. All right, next one, needed in small amounts. Enzymes are not used up in a reaction. This is an important one. Basically, what it means is that after the reaction, the product, you'll still get the enzyme back. So they are not consumed as a part of the reaction. They just help in facilitating. Not used up. They work by lowering the activation energy. So another important point, activation energy is the energy required to start a reaction. So they make the energy is lower, and you can say that that lower energy, they are doing the work for that lower energy. Next is they're very, very selective about their substrate. So this is an important one. Plus, it is going to make more sense when we look at some images or diagrams from your book. There are factors that affect the enzyme function. What are those factors? They are pH, temperature, concentration, inhibitors, and activators. We'll take a deeper look at them. And then some enzyme need cofactors for them to work better for metabolic pathways. So here you can see a diagram of activation energy and how it's different when we are using a catalyst or we are not using a catalyst. So the red dotted line is with a catalyst, as you can see over here, that the activation energy has considerably decreased as compared to when we are not using a catalyst. So this is the activation energy when we are not using a catalyst, the purple line, and then the red dotted is the activation energy is lowered, although they are beginning at the same level, the energy is at the same level for reactants and products. So on this slide, we will continue the characteristics of enzymes, and remember, we are highlighting the important word. So the next one is that enzymes have always will have suffix ase, ase in the end. That tells us that they are an enzyme. After this as is an example. An example is, for example, sucrase is an enzyme that helps in breaking down sucrose or digesting sucrose, and it breaks down as glucose plus fructose. Now, when a picture or diagram is being shown to you, that this is an enzyme here. So This enzyme basically has different little shape. The shape here is, this is called the active site, where the substrate is sucrose here, which is going to actually come and fit into this active site, get digested, and separate into glucose and fructose for further use. So now we are looking at some basic definitions that are important in relation to enzymes. Active site is the shape that can fit or bind the substrate, as you can see in the picture. Substrate is basically sucrose here, which is the reactant in a chemical reaction, which will go and actually fit into the enzyme. And then let us draw a picture for enzyme substrate complex. So for this given reaction here, what will basically happen is, let us say this was our enzyme and it has that specific shape. And now we'll have the, it's not a perfect drawing. I'm sure when we have that picture or image from the book, it'll make more sense to you. But the enzyme has now, the substrate is bound into the active site. So this is the, you can call it substrate. And that's how basically, this is the enzyme substrate complex. So this whole piece is called the enzyme substrate complex. Now, this is a better picture of what we just did on the last slide. Here, as you can see on the last slide, we have taken a look at substrate, active site, and the complex that results. The same thing is explained here in a better form to you, where they are showing you the active site, labeling the substrate, and then enzyme is changing shape slightly as substrate binds. So it's actually binding it, helping in digestion. And as you can see in the end, the products are leaving the active site of the enzyme, and then the enzyme is still there that can be reused now. Now on this page, we are going to take a look at enzyme models as to what are the two main models that are available or that are to be seen. The first one is lock and key model and the other one is induced fit model, and we'll take a look at definitions of both of them. So the substrate, when it exactly fits, so that's an important word, where it exactly fits into the active site of the enzyme, like how a key has, every lock has a specific key, so you cannot use keys in other locks. They just go to their specific lock. So in the same way, the shape is like exactly replica of what the substrate is going to be. So let us say the substrate has this shape. It is going to be exactly this shape that it can fit the substrate into it. Now, the induced fit model is a little different than lock and key model. Basically, the different part is that as the substrate enters the active site, the shape of the enzyme changes. So these are the important words, that the shape will change so it can actually bind the substrate. So it's not the shape is not initially exactly same, but it changes a little so it can bind the substrate into it. So we will now take a look at the factors that affect the enzyme activity in the body. So the very first factor that actually affects is the pH of the body. So the first point is that enzymes function at a certain pH. So they are very, very particular about the pH value, and lower or higher pH value can actually affect the function of the enzyme. Now, the important part here is that the pH that they work better at is between 6 to 8, because the human body also works well at between 6 to 8 pH. Now, we are going to support our answer with a graph here, where on the graph on the y-axis here, we are showing the rate at which the chemical reaction is occurring. And on the x-axis here, we are showing the pH value of the reaction. So you can see that at pH 7, basically the rate or the speed of the reaction is at its best. Now there are exceptions to this rule also. Pepsin is a special type of enzyme that is found in the stomach with a pH of between 2 to 3. So there is an acidic environment in the stomach, and that's where this enzyme is found. Next, changes in pH can make or break the chemical bonds. So when the pH changes, what happens? The chemical bonds are broken, and it's actually amino acids, right? So what happens is they become misfolded, and then they cannot bind to the substrate that well. So the pH has to be maintained for enzyme to function properly. So another factor that affects the enzyme activity is temperature. Now, enzymes need a specific or optimal temperature to function perfectly. At a certain temperature, the kinetic energy increases, molecules move really fast, so the binding is highly likely. So that's the important point. And then because kinetic energy increases, they are moving faster, and which helps in binding. Next up is the graph. So in graph again, excuse me, here we have on the y-axis, we have the rate, and on the x-axis, the temperature. So at a certain temperature, the rate is at its peak or highest. Now, if it's too hot, the enzyme can get denatured. Denatured is basically the word that is given to it becomes misfolded, and it cannot have the substrates cannot bind to the enzyme. The important piece next is that the optimal temperature is 98.6 degree Fahrenheit or 37 degrees Celsius, which is also the normal temperature of a human body. Now, another important factor that affects the enzyme reaction is going to be concentration of the substrate. Now, there are four important points to remember here. When the enzyme concentration is low, the reaction is slower. When the concentration of enzyme increases, the reaction will be faster to a certain point. So it basically means that we cannot have more of enzyme, and that will tell us that it's going to make the reaction faster. It has to be optimal. The concentration has to be exactly optimal for the reaction to be faster. Next is substrate. Substrate is basically the molecules that are going to bind into the enzyme. So if the concentration of the substrate is low, the reaction will be slower. And then if the concentration of the substrate is high, the reaction will be faster to a certain point. And then we have the graph supporting the same information. All right, so another factor that affects the enzyme activity, they are called inhibitors. Now, the word inhibit means to slow down. So these are basically, they will actually stop or slow down the enzyme activity. And there are two types of inhibitors. The first one is competitive inhibitor, and the other one is non-competitive inhibitor. Visuals will make it more clearer. So basically, in a competitive inhibitor is they are very competitive in nature. So they are competing with the substrate. What happens is, let us say we have the enzyme here. So there is going to be a substrate that is going to come and, basically come into this area that is going to be the active site. But there is another piece that comes into that place, and it will come into the active site, and it will inhibit the substrate to come. So the inhibitor is shown in green here. So this is going to be the inhibitor. And because it's going and, actually binding into the active site, they are called competitive inhibitor. So now the substrate, once it is, here, the substrate won't have a place to come and bind with the enzyme. On the other hand, the non-competitive one basically are going to be, let us say we again have that enzyme, and they are going to actually find a place. Again, the non-competitive also I'm showing in green, and they are going to actually come and bind themselves in another place, not in the active site. So what basically happens is that the enzyme is going to change shape now because of the inhibitor that is here, the non-competitive. Hope it makes sense with my diagrams here. Now, because it has changed shape, it is not able to have the substrate. Substrate cannot come. Now we are going to take a look at some examples of enzymes and what basically the reaction, what is something that they break down or help in digestion of. So the very first one is lactase. Lactase breaks down, it is an enzyme that helps to break down lactose. And what does it break down lactose into? Glucose and galactose. So next is sucrase. Sucrase is another enzyme which breaks down sucrose into glucose and fructose. All right, number three is going to be amylase, breaks starch. Lipase breaks down fats. Pepsin, that we already talked about earlier, also breaks down proteins found in the stomach. pH value is between 2 and 3. Protease breaks down proteins into amino acids. Number seven, isomerase. So with isomerase, basically you just have to understand the word isomer. It helps in catalyzing the rearrangement of different isomers. All right, a couple of more examples of enzymes. Kinase, it basically helps in transference of phosphate group from ATP. Remember ATP where we had three phosphate groups attached? So it helps in taking that phosphate group to other molecules. Dehydrogenase, so dehydrogen removes hydrogen from atoms, as the name says. Hydrolase catalyzes hydrolysis, so helps with hydrolysis. The name actually explains that. All right, so this is an exercise for you. You will be actually writing these questions and answering them, 1 through 7. The information about all of these questions has been discussed in the video, so feel free to go back and refer to the information. And I'll see you in my next video. Thank you for watching. Have a good one. Bye.